So variation. Variation became one of the most important aspects of modern biology. I think you are aware of it. And when I say variation, despite the fact that we will focus on human variation, I should emphasize that it's far from being exclusive to human variation. I must say that a lot of the program and tools that have been developed had to be adapted to human because the scale of the variation is enormous, and a lot of these kind of visualization tools and so on cannot hold it anymore. You know, yesterday I searched for, and in many of those places it says not anymore presenting because of the amounts. I mean, so there is a need to build a new way to present human variation because of the scale. But otherwise, I think it's a very important topic. I could not resist then putting this slide that tells you, everybody I guess understand what it is. This is the corona, not the beer, but coronavirus, which is this spike was mutated recently, and this mutation led to this viral, I would say, epidemic. I don't know if the name epidemic holds, but to the fact that more than 100 people have died already, with about 5,000 reported infected. Probably you can put another factor for the real numbers, but anyway, what it tells us that it's all about a very minor, almost minor variation in the virus that moved from probably being infecting, we are talking about this guy, by the way, infecting bats and all kind of ferret and so on, into jumping to human. And really it's a beautiful exercise, but not for next week, really looking at it and understand exactly, basically, what happened in this very quick evolution, which is really the basis for variation. So this is just as a motivation. So those definitions we already discussed. I just want to mention that when we discuss the reference genome, this is a slide taken from that, you know, in the beginning of the class. When we discussed the reference genome, we said we have to use some dictionary to understand what we are looking at. So the term reference and alternative, major and minor, and so on, have been used already. But remember that what I'm trying to do today is to move it from a single genome, in which you can have one or the other, into a more population view of the variation. So this small statement that we are moving from one genome into multiple genome must use different terms, such as frequency and allele frequency and so on, and that's what we are trying to move. So I'm pushing this limit, although the term haplotypes and LD comes back, of course, in the population level. So the one thing that I think is become very important and was almost overlooked for many years is this aspect of private mutation. I'll say more about it, but private mutation are mutations that have not been fixed in the population like we studied in classical evolution, but instead are kind of private. Private means your own sub-branch of the family, you and your kids and so on, but not really part of the frequency within the global population, and those numbers are enormous. I mean, it was completely unseen, I would say. And of course, the structural variation that I already discussed become much more important than previously anticipated. That we are not the same in terms of structural variation. When I say structure, I mean not single nucleotide change, more than that, okay? So somatic germline we already discussed, coding, non-coding we already discussed, and this is just a reminder that when we talk about the effect of mutation, we can think about the functional, the endpoint on the functional of the protein, what happened if we are talking on coding genes, such as synonym, missense, which is change of amino acids, And the most, I would say, devastating, usually, not always, by the way, event, which we called loss of function or nonsense, which is stop gain in the gene. Now, although, you know, like from the first order, I would say that nonsense, loss of function, are the devastating and carries a lot of this disease story. It's not always the case because of the type of inheritance and the fact that sometimes when you have nonsense, there is an activating mechanism of NMD which remove this and kind of don't let this bad product be dominating the cell or the system. So to those that missed this comment, let's move on. So just a way to show the numbers. All biology talks about this, right? Most biology, 20,000 genes. But we have another 20,000, at least, that are non-coding, that we understand much less about. We have a transcript, which is 4 times at least the number of genes. This is all the alternative splicing and so on. So this is the real number of entities in the cell. But this is the number that we are accumulating in terms of variants. So you see that we are in different scale, no matter what. And those variants are really in the many, sometimes currently the number is like 100 million variants that are known and millions of structural variants, millions. So see the scale, and this is just from ensemble from 2017, and the number keeps going. So one way to look at the variant is by the table of variant, and this is just a snapshot of the gene view. And I want just very quickly just to show you when we talk about variant, what are we talking about? So the most important part is where, location. And of course, as we already discussed, it's important to know which reference are we talking about. Is it HG19? Is it the 37, 38? Okay, so this is one. The second is the identifier in the SNP-DB, which is RS something, but not always you'll have one like this, definitely not in the private mutation. Then whether it has a heterogeneity or homogeneity, so 0 means that there is no case in which the mutation is, the 2 variation has been shown in the same individual. Validation means how do we know that this is the case? And there is a logo for 5 or 6 resources. 2 of them we'll talk today about. One is the 1000 genome, and the other will be the ExAC. The 2, but there are more. I want. Then, very important, the minor allele frequency. As I said, it's not whether you have it or not. What is the frequency in the discussed population? That's very important. Then, this is then whether there is a 3D or not have a 3D. The function, whether it's synonym and apparently not affecting, although it's a bit naive to say it, and the missense, which is affecting, so you see it. Which one is it? What happened to the amino acids? Where in the codon it happens? The amino acid position in the protein and so on. So you understand all this information tell us already something about what is going on. The one browser that I'll just give one slide of this, and on purpose I'm not illustrating it because I'm not so much fond of this browser, but it has few features that are interesting. This is the 1000 genome browser that was dedicated for this. So I just want to show you what you get. This is an exon, as you already know. This is a pretty nice feature in which you can jump from one exon to another just by clicking. So it's jump over all the intron, which is pretty good feature to use. And this is selecting of exome. This is to say by color whether it's a pathogenic or not pathogenic. So immediately you see a patch of pathogenic. So I'm saying, you know, as we already discussed, the browser has many features, but the most important one, in my view, is that in one view, you understand the story without getting into the table. So this is a good way to show it, the pathogeneity. You have here also what was not there before in the last version of the 1000 genome, the structural variation, which was, as I said, below the carpet. I mean, no one talked about it. And you have here the minor allele frequency of the 1000 genome. So this is the browser. This is just another way to look at the browser. And you can look at it through the variation viewer. You have many filters, as we discussed in the CleanVar. When I said many filters, you can say, OK, show me only things that are sitting within the dB of SNP. Why? Because you want something stable and not falling into maybe a mistake in sequencing, right? So you do this. You can say, do you want it in CleanVar or not? So CleanVar, sometimes it's independent evidence that it was shown. So you can only look at this, whether you want pathogenic or risk factor and so on. You can click and have a subset of the variation according to your filter, and the filters are very good. So this is what I like in this 1000 genome kind of presentation. And in the end, you can go always to the SNP report, and the SNP reports will tell you exactly location, organism, the allele, whether it's a SNP, it's a SNV or other type like indel and so on. Immediately it tells you the frequency in ExAC. I'll go into it. For example, this frequency has been seen 1 out of 120,000 allele that have been checked. Okay, so you understand that it's extremely rare, but it happened, and I'll show you this, and immediately all the association with the different, I would say, version of the human genome. Okay, so remember, nowadays, almost all the information that is accumulated from now on is based on NGS, which was not the case in the beginning of the project. Now, NGS, while it's fantastic, has its own kind of pitfalls. I won't go into it, but let's say, just for the sake of statement, that there is no mistakes. Everything is perfect, which is, of course, underestimating the true mistakes of the sequencing. We still have this big question, whether a variation that we see is just there because of, you know, a story of evolution, because of whatever, or is it causal? This is, I would say, one of the most challenging and unsolved question in the field of, genetic association, the genetic causality. Now, what we see in this NGS is like this. We can see a person in which we read many times the same nucleotide, and you can see that it has 5 C and 5 T's, right, in this position. But of course, I don't have to tell you that when you look here, you see only 3 T's, so your statistics is different, right? So be aware that when you see it, there is underlying statistics behind it. And of course, the challenges are many of what I just said, the mapping, the mistakes, the base calling, what algorithm was used to make sure that we know what we're talking about, and so on. In general, in the clinics, there are basically 3 lines of people that are using either all genome, in which you get most of the genome. It's very costly, and you cannot get a really, really deep depth. The alternative will be the all exome, that I'll talk in a minute about it, which is covering only 1.5% of the genome, however, at a reasonable depth, so you're more reliable on what you see. The cost is somewhat in the middle, and what is very commonly used, which is panels, in which you just select what you want to look at, which is, of course, not cost, the cost is low, and the coverage can be as high as you want. So very reliable. This is the way to do it. So what you see here is an NGS of any kind. And the one important thing is that sometimes you don't want to look at anything unless it was read enough times. Okay, so you put some thresholds saying, don't show me if it's lower than that. Not because it's not true, but I cannot do the statistic, I cannot rely on it. So some threshold is put, and then it goes directly to the resources. One of them is the 1000 genome, which is really very creative when it started for the saying, and for the two reasons. One, this reference genome may be far from representing the population, which is true, of course, by definition. Second, maybe we have a very biased view and we do not look at the diversity of the human population, which was very true. I mean, African, Chinese, I mean, they were not even part of this initial project. And that was what's going on in the human population. And third, can we devise some kind of a more statistical based probabilistic tools to understand what we see in terms of population? That was missing completely, and that was the goal. And the goal, what I show you here, is how it was evolved from let's say 2000 and something to 2013, that the project was shut down. I mean, shut down, namely completed, at least for the goal that were. And you can see immediately by the color is the different ethnic group that were accumulated. So a priori, they decided to go to quite a lot, like 21 different ethnic group, and it's really super interesting to look at it, at the difference between the different groups. So what we learned without knowing is that in the end of this 1000 genome project, plus minus, every individual have 100 a new de novo variant within, I mean, from parents to kids. So I'm mentioning this number not because it's a magic number, but because think about it, what happened when people are doing whole genome or whole exome and they end up with 100 changes. So which one is the one that causes a disease or some phenotype and so on? The question is not trivial despite the technology. So this is really to remember, because a lot of people think that once they do parents and so on, there will be one new de novo mutation, everything is clear. This is not the case. Okay, this is just a comment about the practicality. One very strange figure that I kind of like is the fact that from the beginning saying 200 people to the end, which is now, which is 2,000 people, despite the fact the nice number 1,000 genome, which is 2,504 people actually, but the name remains for the project. You can see a few things that I kind of like. First, that the number, this is the number of the number of variant is still getting up. I mean, more variants are added to the list, but more interestingly, the number of variation of indel became, you see more and more indels. So you can see that the indel number, length and number is growing. So you see more variation while you sequence more and more versions of the genome. This is something that is kind of nice. Okay, so I'll skip this, and I'll just say, maybe you look at it later, but I'll just say that there are some really interesting findings that we didn't know by looking at many genomes. For example, some regions have a huge amount of variation, like the HLA that is responsible for the immune system. I would say that no one knows what to do with it, so they put it aside in many aspects, or they do some heuristic very naive. So this is an area of research that is phenomenal. I mean, understanding this. Subtelomeric regions are changing like crazy. There are regions that are fixed, nothing happened to them. Why? No one knows. There are this issue of imputation that needs to be added, and the haplotypes that sometimes are not phased, so you just get a punch. You don't know if T and G are in the same strand as G and A, or it's just a mix, because you read the diploid site, and it's not phased. You don't know exactly that this goes always with this, and with this. So this is the haplotypes issue. I should say that I think I already mentioned this variation at the population level, and I want to take you to these two solution of the variation in which the sequence variation estimation can be done by kind of guessing what's the mutation rate. What's the mutation rate? We know the size, so how many we expect to see. And indeed, we expect to see quite a lot. And this is, initially, it was one SNP per 100, but now with the current big genome project, we know that basically we have one SNP, if I include the private, every 8 nucleotide, which is unbelievable. Every 8 to 10, there is somebody in the population that has this variation. So basically, it's unbelievable number, right? But those are not the common one, but the rare one. So this is a big difference. This is the snapshot from the 1000 genome in which they try to cover, as I mentioned before, many of the population. And of course, the idea is that many of those variations will have a very low effect, but they're accumulating a contribution to some phenotype, like height, like weight, like a tendency for cancer or whatever, okay? So this is the one approach. Of course, it complements with the other very known classical approach of family business, in which, of course, in the family, one can understand that there is mutation that maybe have a high impact, but the scale is much, so it's a single gene with major effect. This is the family story. And now the field is moving into a population story. Okay. Two words on history. The project has been moved to 2013, but from this time to this time, and it was broken into 3 phases. The phase 1 was the initial round of exome and low coverage. Low coverage means only to know what's going on, of 1,000 individual. Then phase 2, there is no very important addition, but phase 3 ended up and stopped by having over 2,500 individual and created the new catalog. Now the format of the catalogs, you are familiar with many of them. The VCF, you are going to talk about, so I'm skipping this. But what I'm saying is that the population level have been decided by saying, okay, in this population, 1% will be some kind of a standard to show the difference between, of course, abundant to non-abundant. But of course, it's a very arbitrary. I don't think a lot of people are using it anymore. So think about it as a minor allele frequency being anything between 0.0001 to whatever, to 50%. Okay. This is the combination of people, just to show you that people from Europe, from Africa, from Japanese, Chinese, and so on, rather quite balanced, I must say, which makes it even more interesting. And again, it started with few hundreds, but now the number grew up. And within the same effort, they did some trios. So to understand really the parents-child differences, and to basically at that time, even to confirm a stability of sequencing. So it was for the both reasons, to show that, as you can imagine, if you have mistakes, it won't be explained by parents-child kind of information. And this is just the growing of the phase of the, and of course, two things that I want to mention is this term that's called tag SNP. Tag SNP means the SNP that once you take it, represent maybe 3, maybe 10, maybe 20 SNP around you that will behave identical because of the recombination patches within heritable movement between generation. I didn't explain it well enough, but I think the intuition is very clear that when you do recombination, you don't do it at a single level of variant, but at a patch. And those patch the representative can be the tag SNP, and the other are the imputed or the rest of the SNPs. Okay. So just maybe I want to do maybe illustration for the exact. Okay, I'll take another 2 minutes. So there are 2 ways to do to get a combination of variants. One more classical one was on a chip. So on a chip you can put kind of hundreds or 500,000 of probes and ask specifically, this person, does it has it or not? To this aspect we call genotyping. Why? Because a priori you decided what type of SNP are you looking at. Now with this method you can never find new stuff. The alternative is what the Illumina was doing, which is to sequence. Specifically, the most, I would say, advanced project are only the exome, this 1.5%, right? And then you have all the information that you want. Either this is still in the type of genotyping, but we can really read the reads and get exactly all those new private new mutations, right? So that was the initial idea of the 1000 genome. I'll just skip this, and the goal, I think you kind of understand. This is just the history of how many SNPs were accumulated so fast from the beginning to the end. It's really very, very amazing speed. Remember something that I don't think many of you are familiar, that initially when they started, the reads number length was 35. Now in 35, you cannot really read a deletion of 200 base pair. So it's useless, basically. This is why when I say the structural variants came much later, it comes with the technology, because this technology failed to do any reasonable structural variation, okay? And that was the standard. So this is why I'm mentioning it. As I said, the browser and all the information is in the 1000 genome. I just should say that even this project showed that many of them were not even in the SNP database. So they were new to the SNP collection catalog. Yes?
How long is the current read length?
100. 100 from two-sided. So each probe is at least 100, but they do something that is called paired tail, so you know the difference between the two. So you know exactly what is the length differences. So you have a very strong, and actually the structural variation is done now differently by a chip that was designed for that. So they split the technology, because this technology is very good for small reads, and the other technology is better for, so it's 2 chips, but this is a kind of minor. This is a very nice picture, just to say that this is the different ethnic group. This is the number of allele counts. This is in millions. And you see clearly 2 populations. This is us that are very kind of down the hill. And this is the African, which is really have huge amount of variants, because this is the source. So it's very nice to see also population. This is the number of people that are having 4 million, and those are having 5 million, and so on. So it's very nice. This is really from the last paper of the 1000 genome. Everything is ready. There is a very nice, kind of a video that explains the history, so I'll skip this. Now, just to remember, going to your question, sometimes we consider all this technology, okay, NGS, who cares? But this is just to show you that it is done by PacBio. PacBio knows how to read thousands in a row. So PacBio, and only this technology, could show this inversion, can show this deletion, huge, can show this complicated story. You cannot do it otherwise. So you understand exactly why, because you see it in a one string. So you understand. What you see here is really the dot plot that I think I mentioned in the beginning. And this is the array that supposed to show it. Now, ExAC, very short, in few words, ExAC was initiated in 2016, basically after the 1,000 genome. And the idea was the following. Let's take 60,000 healthy people, not related to each other, so representative of 60,000 people, and do full exome sequencing, not genotyping, sequencing. And doing that, and at the same day, expose it to the public. However, because of privacy, you cannot expose the individual, but you expose the summary, statistics of everything, divided by the ethnic group. So basically 60,000 variants, people, means 120,000 allele. That was the number that I showed you in the beginning, 1 out of 120,000. This is the ExAC. Now, the ExAC really changed completely the way we understand human population. Why is that? Because a lot of private mutations have been identified, huge amount, really. It means that whenever people are doing somatic sequencing for a cancer, you need to go through your own basis of a variation. Otherwise, you believe that this is a variation that happened due to the cancer, but actually, it's your own germline variation, right? This changed completely the field of looking at variation on an individual level. You understand this? I mean, this is very important. This is why ExAC is a really phenomenal project. Basically, it was canceled as a standalone, and now it's part of a bigger database. It's called gnomAD. And I'll just skip this, but this is just a summary of this ExAC that you see in each person, really, 500 rare, missense mutation, 100 loss of function, 5 disease causing, 1 de novo mutation. I mean, really enormous diversity that no one really appreciated before taking the population that are healthy population and not related, so you don't have many families that are within. This is very nice, and this is just to show that, again, European are the majority of the 60,000. It was done in the US with not that many of South Asian and so on. This is the ExAC consortium, and really beautiful, and each person provide these 2 allele in each side, and as I said, every 8 nucleotide you have some variation. So gnomAD is the replacement of the ExAC database, and of course you can learn a lot about disease, penetrance, evolution, diversity, and so on, despite the fact that it's all aggregated. You cannot go back to the individual, okay? This is the payoff of having this only summary, so you cannot really say that this person have both this and this and that, okay? So you just have a summary statistics. And I think I'll stop here. Maybe should I take 2 minutes for demonstration of the gnomAD? I think it's very nice. How many of you are familiar with the gnomAD database? Sorry, no break now. No, it's a very really important and good resource. So I'll just do it in really short time. Let's take this. This? Oh, this, okay. Okay, let's do gnomAD. Okay, so gnomAD has changed only very recently. Can you see it good enough? Only recently, by showing that it has 2 versions. So let's look at the initial version, and ExAC is is hidden here. So you can search by gene, by region, by variant, by whatever you want. Let's take a, I don't know, a gene. Okay. So what you see here is I'm looking at the gene viewer. So, and I'm looking at ExAC. I won't talk about, okay? So I'm looking only at ExAC. As you see here, the ExAC covers 60,700 samples. And the other, for example, gnomAD v3, version 3, has already 71,000. And gnomAD, this one, already have 140 people. So be aware that not all combination is so good for you. So if you are solid, go to ExAC. And the ExAC have exactly the reads of each and every exon. Now, you see the viewer of the exon, 1, 2, 3, 4, 5, 6, and so on. And each exon, as you can see, has another 75 nucleotide from both sides and nothing else. So the intron are gone. Okay, no intron, no story. All the story is on the exons. And what you see here is something which is very nice. First of all, the ClinVar, whether there is any information in ClinVar. And you can say, show me only the missense. Don't show me the synonym. Don't show me any other. Don't show me any stop codon. Show me only the missense. And immediately in the table, you'll get the table of the missense. And it's, you can immediately say, show me who is the most, let's say, abundant allele frequency. And you can see that out of 110,000 allele, 79 has a missense of proline at position 99 to Glen. Okay, so immediately you can see whatever you want. You can see whether you have homozygous, and immediately you see that, for example, this missense probably is not harmful. Why? There is 4,000 homogeneous, I mean, people that have both allele changes. The frequency is 30%. So 30% of the population has a serine to glycine change. And the source is ExAC. E means exact. And if you want to have something that is more elaborate, you can go not to ExAC, but to, let's say, whatever, this gnomAD. And immediately you see that it's not only in ExAC, but also in the 1,000 genome. The G is a 1,000 genome. This is the ExAC. And immediately you can see how well you can enrich the list of variation, and you can sort them by allele number, by allele frequency, by allele count, by homozygosity, and so on and so forth. And it's extremely, extremely useful. I should say that most people in the consulting, genetic consulting, are using this one, exactly like you. So this is the only source for genomic, I would say, decision in view of population. Okay, so I think I'll stop here. And it's really, there are a lot of very nice options, both visual and analytic, within this very fast evolving resource.